testing has really revolutionized the diagnosis of rare disease. And I know that's a lived experience for many of you in this room. And the take home from that is that we're now in a position where we can use these approaches to provide a diagnosis for about half of patients, a definitive genetic diagnosis for around half of individuals with a suspected genetic disorder. And that's really a great thing. It's something that we've heard that's 5 times better than what we were able to do in the past. And that's something that should be celebrated. But at the same time, it begs the question, what about that other 50%? And that other 50% is really the focus of my research. And it's about, and so what I and my research group is trying to achieve is trying to improve our understanding of the genetic basis of disease, particularly in those specific disorders that my group focuses on, but also improving methods and our ways of analysing genomic data and genomic samples to allow improved diagnosis and then be applied more broadly across rare disease. So if we were to take an example of an individual who, let's assume, does have a single gene genetic disorder, and that they've had genomic testing and that failed to reach a diagnosis, and we ask ourselves, why might that have testing missed that diagnosis? And Sue sort of more broadly went through some of those causes that might explain that. And the 2 I'm going to focus on today is the cases where the affected gene in that individual has not previously been associated with that disease, and therefore the clinical testing provided cannot provide a result because it didn't even know that that gene was associated with the disease. The second case is where the disease-causing variant was detected by our technology, we saw it, but we lack the knowledge or the understanding to actually associate that with disease. These are the cases, the variants of unknown significance that David touched on, and they're a real challenge across the field at the moment for how do we do a better job of identifying true pathogenic variants out of so as a researcher, where I come in is I work with families who have gone through clinical genetic testing and did not receive a result and had a negative result. And then working with clinicians like Sue, they are recruited into research studies. And it's at that point I become involved. So I actually have no direct interaction with families at all, which is a bit of a shame. But my focus is really on analysing the data. So in our group, if we were to take on the analysis of an individual, what might that look like? This is just a really high level overview. But essentially, our first aim would be to get the information from the clinician who'd recruited into the study, a little bit of information about the presentation of that case, and either access to the genomic data or access to DNA samples. We then want to sequence, preferably the genome of that affected individual. And in my research group, we really like to work with trio data, so we want to sequence the DNA of the parents as well, and then analyse all of the variants to identify and all 3 of those individuals together. First thing we can do is things like exclude variants that are common in the general population, under the assumption that if a variant is common amongst the general population, it's highly unlikely to be associated with disease. And so you can think of these as not spelling mistakes, but perhaps just differences in spelling, American English versus English English, Australian English. And then we'll go through a process where we will try and prioritise the variants and select the ones most likely cause damage to a gene or be associated with the disease. These are the filters that David was talking about earlier, and they help us go from a very large number of variants to a small number of variants, but also the step that we can get wrong, and we spend a lot of time thinking about. If we're lucky at the end of that, we might identify what appears to us to be a damaging variant in a gene that could plausibly cause the disease that we see presented in that individual. If that gene has not been associated with disease before, the question becomes, what then? At this point, this is what we'd call a suspicious candidate, but really we can't do anything with it. At that point, we're generally left with 2 options. Option 1 is what we'd call, using loosely called functional studies. So this is where we'd go into the lab and design some experiments that are designed to actually understand the effect of that variant. So see if we could prove that it actually changed the function of that gene. Sometimes that's easy, sometimes that's pretty challenging, depending on the variant and the gene that we're in. Second option is find more cases. Can we find more unrelated patients with a similar presentation that also have damaging variants in the same gene? Really, ideally, in the best case scenario, we use a combination of options 1 and 2. So I'm just going to give one small example of that from work that was done partly by my group and published earlier this year. And within my team, this work was actually led by a PhD student in my group, Guy Hellman, shown here in the upper right corner. And this story starts with 2 unrelated patients that were part of a group of, a cohort of patients that we were studying. And they both had a disorder called a hypomyelinating leukodystrophy. So this, put simply, was that the white matter, which is an important component of their brain, did not develop properly after birth. We performed genome sequencing on these 2 affected individuals and their parents and analysed that data, looking at all of the genes that were known to be associated with this group of disorders. We weren't able to identify any compelling candidate variants, and so then went on to look more broadly to look across the genome to see what we could find. What we found is that both of these individuals had a de novo, so this is a new spelling mistake, in the gene TMEM63A. We went and looked at what was known about that gene, and the answer was really not much. About all we could say about it is that it's highly conserved. So all animals and even plants have a copy of this gene, and it's very similar across all of those species. And that was a clue to us that it was an important gene, but we didn't know much more. So our first question was, can we find more patients? Luckily, we now have available to us tools like GeneMatcher. This is a service that David referred to earlier, where researchers and clinicians can put in some basic information about the gene they're interested in, and the presentation of the patient they might be working with, and be connected with researchers and clinicians from all around the world who might have similar findings. And then it just allows a matchmaking service so these people can get together and talk to each other and see if they are actually working on similar things. To give you an idea of how international an effort this is, these 2 patients that we're working with were actually identified by a clinician called Nicole Wolf, who's been a long-term collaborator of ours from Amsterdam. She provided us with the genetic samples that we were working on here in Australia, where we identified these, did the sequencing and identified the candidate. And through GeneMatcher, we were put in touch with a group in Canada who also had 2 patients with de novo mutations in the same gene, TMEM63A, and they had a very similar presentation to our own cases. And it turns out that those 2 cases were actually recruited as part of a cohort that was developed in China. So where we were at the moment, we had 4 patients, all with a very similar presentation, 3 different variants in this gene TMEM63A. And the question is, was that enough to get us across the line? Did this give us proof that this was a new disease gene? And the answer is almost, but not really. It was right on that borderline of what would be acceptable. At this point in the study, we were quite lucky that a paper was published by a group from the Scripps Institute in California, who had been working on the biology of this gene TMEM63A. They weren't interested in the clinical side at all. They were actually just trying to understand the fundamental function of the gene in our genome and what it did. What they were able to establish is that this gene is what's called an ion channel, which we mentioned earlier on. And you can think of ion channels as being like a gate in the cell membrane. So, and what they do is normally that gate is closed, but if it receives the right stimulus, the gate will open up and allow ions to pass through the cell membrane from the outside of the cell to the inside of the cell. In this particular case, TMEM63A is a pretty special type of ion channel in that the stimulus that causes that gate to open is actually mechanical stress to the cell membrane. So basically, if you take the cell and you stretch it a little bit, you try and stretch the outside of it like a balloon, that causes this ion channel to open. It's one of only 2 different families of ion channel plates in the human genome. So the stretch causes the ion channel to open. Not only that, this team developed a nice little assay where they could do this in the lab and actually show that this opening and closing effect. And what they were able to do for us was say, compared to controls, so that don't carry any of these variants, showing how activity of this gate, they were able to show that our 3 variants have basically no activity and this gate stays closed, proving that these variants do actually damage the function of that gene. So altogether, that gave us 4 patients, 3 different variants, and the experimental evidence that these variants do damage the function of the gene, and that is a compelling package which is enough to publish and put out in the literature and show that this is a novel disease gene. I also just want to make the point that while some of this work was done in my lab here in Melbourne, this is really a large international effort, and just the number of people listed on this paper give you an idea of the size of the teams that are required to bring together the science and make these discoveries. Next, I'm just going to quickly touch on the second example we talked about here, which are these variants of unknown significance, and these ones that can be hard to identify simply by looking at the spelling mistake and deciding whether it might cause damage and be disease associated or not. Again, I'm going to just give an example from one family from one of our studies. And again, this was a hypomyelinating leukodystrophy, a common theme in my group. This was one male child that we started working on this case way back in I believe, late 2012. And we sequenced first the exome and then the genome of this child, trying to understand the cause of the disease. In this case, he had not only a hypomyelinating leukodystrophy, but also an unusual and specific bone growth disorder. And these combination of symptoms together was very unusual and actually only been reported once before. And that was in one family where still didn't have a genetic diagnosis, but they were able to see that the only males were affected and it was inherited through the maternal lineage, suggesting that the damaged gene was on the X chromosome. So we looked really closely at the genomic sequence of the child, and then also paying particular attention to the X chromosome, and we found nothing. What we didn't know at the time is this is something we'd overlooked. So there's a gene on the X chromosome called AIFM1, and this is just a picture of the full length of that gene. And if we zoom in on just this one little part of that gene, this is one exon, and in our family, they had one spelling mistake here, a C to a T change. But this type of spelling mistake was one of the ones that we thought, that didn't actually change the encoded protein at all. So we thought it was safe to ignore because it was assumed that wouldn't actually change the function of that protein because the protein wasn't changed. And so it was one of these things that was filtered out by the filters we were using at the time. We continue to try reanalyzing this family for, I can't remember exactly how long, 2 or 3 years without making a whole lot of progress and got nowhere. Until through our networks, we became aware of several other families that were being studied by several other groups around the world, and they were all in a similar position. They tried doing the analysis themselves, and all of us had come up with a drawing bank. Luckily, this group of people were collaborative enough that we decided what we needed to do was bring all of that data together in one place, and hopefully together we could make sense of it. I was fortunate enough to be entrusted with that task and was sent variants from several of the other groups where they were working with similar families. And what became very obvious very quickly to us is if we look at that same region, we were able to identify similar, very rare variants, all located within a few base pairs of each other. So these were just all right in the same region with the same gene. Similar to our variant, most of these variants were changes that we thought were probably not likely. If we look at them without knowledge of this, we'd probably decide they weren't likely to impact the function of that gene. And so they could be easily overlooked. But together, we can see it's a compelling picture start to come together. And we were able then, once we had this information, to work with other collaborators to go back into the lab and again show that these variants did have a drastic impact on the function of the gene AIFM1 and were the cause of the disease. So again, we got to an answer for this. We were able to publish it and make it available to the genomics community and to hopefully enable other families to also get a diagnosis. But this case really stuck with me in terms of why did it take us so long to get to that answer, and what could we do better about it next time? How could we speed that process? Why couldn't we have got that answer in 2012? I came up with 2 main answers. One, that we need better methods for identifying that particular type of variant that was easy to overlook using our current filters. And that's now totally changed the focus of my research group, and we're spending a large amount of time working on that particular type of variant and developing new methods that help us identify them. And we're actually starting to get some good success out of that. The other important take-home message for me was the importance that we need to change the way that we share data. So in this case, we had a great outcome, but it relied on the fact of our personal networks bringing these cases together, that we could bring this team of international researchers together. And that's great when it works, but it's obviously a system that could easily fail. And we actually need formal ways to responsibly and ethically share data from these research projects internationally. So these matches can be made automatically and bring these small cohorts of ultra rare disorders to speed this type of analysis, because that's what we really need to inroads to these kind of conditions. That's all I'm going to talk about today. The take-home messages for me, I hope this gives you some insight into, is that this is a field still making rapid progress. Our technology's come a long way, and that's great, but we're still catching up with that in terms of what we can do with that in the research land. And so that means even someone who has a negative or inconclusive genomic test today, tomorrow there's every chance that we might be able to turn that into a conclusive result. The second is I hope you see the need for this data sharing internationally and enabling that. And it's something that we really need to take on board and explain to research participants what's happening, but it's something that will really make a difference to empower these projects. Lastly, I just want to take the opportunity to really thank all of the families that have participated in the research we do. I know that many members of the audience will have participated in research. And as a researcher, I can't do what I do without families that agree to participate and get involved. I just really want to express my thanks for that. And just lastly, I just wanted to touch on today, there's been a common theme of talking about diagnosis, and even in the research space, I want to acknowledge that that's really just the start of the journey. You guys all know that, I'm sure, but for me, it's something that's important to remember and remind myself of. But it's that first step, and once we know the gene, once we know the diagnosis, we can't start thinking of things like developing improved therapies until we actually have that. So it's that first step in a long road. With that, I just want to say thank you very much for your time.
Thank you for your presentation. You talked about the speed that things are changing and you're discovering new things all the time. What happens when we have a diagnosis or have testing and don't have a diagnosis? How often should we go back to say, found anything new yet? You know, anything? Or can that happen automatically?
So I should have stated up front, I am not a clinician and I'm not involved in the clinical testing side at all. So really, the ideal world would be often and regularly, but that really becomes a resourcing issue to how often we can go back and look at, reanalyze the data, and if systems can be put in place in the future to allow some degree of automation to flag new things. At the moment, I don't think that those systems are not globally in place, but it's definitely something that the field is aware of and want to improve, and hopefully that will be enabled in the future. But in terms of how often, I can't give advice on that.
Just from a clinical perspective, I think Cas has touched on, obviously resources are a big factor. We do generally say to families to revisit in sort of about 2 to 3 years. Because we know obviously what we're going to be able to say in 1 year is probably not going to be huge, but you know, it would be reasonable to revisit with your genetic service in about 2 years or so.
I think there's no one answer to that. So for example, for me, I have no capacity to recruit people directly for research. So if someone spoke to me, I would be saying, talk to people like Sue who can recruit into the projects that I'm a part of. Other people doing other researching specific disorders may have a pathway that if you find out that they're researching a disease that's relevant to your family, you can contact them directly and they might be able to give you advice on. So I think there's no harm in asking the question, but the answer will change depending on who you're talking to.
Hi, Cas. It's nice to hear that you obviously were still passionate about looking for an answer for that child. I suspect you've probably looked at my daughter's genome because we're part of the mitochondrial project at the Murdoch Institute with John Christel.
I see study identifiers. I don't see names or faces, so I wouldn't know.
I'll just say de novo and I'll say CLPB gene. Does that mean anything?
Sorry?
De novo and CLPB gene. No, my point was just it's good to know that you guys are still, you continue to look even if you think that, you know, there's not an answer, that it's still on the radar, because the question that you've asked up there, it's been a few years since I've heard anything since possibly finding a de novo single variant gene. I haven't heard anything for a little while, so it's good to know that, you know, this stuff still continues. But I appreciate that it's all funding based and grants run out, and then you've got to apply for another grant to continue funding and stuff like that. And that's why this takes time. So
yeah, no, absolutely. And I think as a field, we are also getting better at putting the systems in place to make sure even in the research systems, people are not lost to time. We do have the ability to open and review the cases. It doesn't happen all the time, but we're getting better at it. That's it.
Okay, sorry. You talked a lot about working on diagnostics with these variants of unknown significance that you otherwise thought didn't cause anything specific, and that there's implications for treatment investigations too. Does your lab work on those sort of treatment therapy-based research as well?
For me, tangentially through a few specific collaborations, but because of my background and my skill set, we're really focused on analysis of genomic data, which is linked to either diagnosis or understanding of the mechanics of particular mutations. Again, where for treatment, the idea is if we understand that well, we can then inform the ideas behind what people might start with therapies.
Thanks, Cas.